An incurable genetic disease that strikes with almost 100% certainty may have just found its match. And there are a couple things here that I think are interesting and hopeful. Huntington's disease affects 1 in every 10 to 20,000 people in the US and is caused by repeats in a gene called HTT. Most people have 10 to 35 repeats in a section of the gene, a little CAG repeat. But in people with Huntington's disease, that repeat is expanded up to 120 times. The more repeats you have, the more likely you are to get disease. If there are more than 40 repeats, disease is pretty much inevitable. Patients endure a slow breakdown of cells in their brain, which lead to a host of motor, cognitive, and psychiatric difficulties, and ultimately death. And until now, like right now, there has been no cure or treatment, only symptom management. This past week, a team from uniQure shared clinical trial results that their new gene editing therapy, called AMT-130, significantly slowed disease progression for patients. The therapy works by silencing the HTT gene. Without treatment, the HTT gene is transcribed by the brain cells into RNA, and that RNA is turned into protein. But those extra repeats mean it misfolds, causing the disease. The treatment lowers the amount of HTT protein that is produced. In clinical trials, treated patients showed significantly slowed disease progression. And this was measured by a number of precise metrics, but most importantly, it means it gave patients time and quality of life back. It is a small study and it did not stop disease progression, but it did slow it. So as a geneticist, here's what I think is interesting. First, this is a microRNA treatment. The therapy inserts a piece of DNA into the patient's brain cells, and that piece of DNA is transcribed into a really small piece of RNA. The microRNA finds and matches up to the HTT RNA and sticks to it, preventing it from being turned into protein. Second, it's delivered by a type of virus called an AAV, and I was kind of surprised by this at first because AAVs have started to fall out of favor in gene therapies. Unfortunately, sometimes they can cause liver damage, which can lead to liver failure and death. They're great at delivering DNA to cells, but their side effects, though rare, have stopped a number of gene therapy trials. However, this trial delivered them really specifically to regions of the brain affected by Huntington's disease. They drilled tiny holes into the skull and then delivered the therapy by capillary just to those precise regions to try and limit their spread. Ideally, it is a one-time procedure, and I think this is kind of an interesting strategy. Now, this is still a long time away from being a common treatment. It's only in phase 2 trials, the results have only been tracked for 3 years, and it is an expensive treatment. But it is a big, hopeful step in treating a disease that so far we haven't been able to treat.